Very good morning to you students. Today we are going to start the new module, that is ecological statistics. So first of all, I'll share my screen and then we can So this is the last module of our paper, biostatistics, that is ecological analysis or ecological statistics. Actually, to say that we should finish this in just 7 hours, to say that we have to finish ecological data analysis in just 7 hours is a very great injustice, because ecological statistics itself merits an entire 50-hour or at least a 70-hour paper in itself, because it has got so much of depth. This is one part which usually the statisticians do not use, but rather it is purely used by ecologists and biologists. And not only that, many of the theories which we study here in ecological data analysis, that is also very important in various other scientific papers like paleontology, etc., etc. You should please excuse me for the quality of my sound because I'm still recovering. So there will be some problems when I speak. There will be some disturbances. So yeah, so that is why ecological data analysis is very, very important. And you're only skimming the tip of the iceberg, because according to your syllabus, you have to study alpha diversity, and under that Shannon diversity, Simpson's dominance index, Pelews evenness index, Markleaf species richness, and Fisher's alpha. It is not alpha, it is alpha. So this is only, it's like, kindergarten level for ecological data analysis. Nobody does this anymore. We have got so many software. So most of the people just know the basics and they go for the softwares. Actually, when we talk about ecological data analysis, a lot of complicated things are there. Jackknifing is there, modeling is there. That is the level which we are talking about. But unfortunately or fortunately, for your biostatistics, only this much is there. So we all know what alpha diversity is. Alpha diversity, is a measure wherein you try to understand in numbers the species diversity and the richness of a particular ecosystem. So here you're only looking at one particular ecosystem. But when you start comparing the alpha diversities of two different ecosystems or two geographical areas, then it becomes beta diversity. And for beta diversity, you have got only three indices here. That is the Morissetta-Horn index, then Sorensen's index, and Bray-Curtis similarity index. Only that much is there for your ecological data analysis. And from my personal viewpoint, it looks very silly. And I feel very disappointed because you will be only studying this and you'll forget about even this. So when you go out into the field, when you try to become ecologist, you will find it very, very difficult because there are a lot of other things which you actually need to learn. Anyway, now, before we go into ecological data analysis or ecological statistics, what we have to first understand is that what is the type of data which we are going to handle. So unlike all the statistics, the conventional statistics which you learned, the descriptive statistics and the inferential statistics which you learned before in the previous modules, this is something absolutely new. So we have to understand the type of data which you'll be handling and which we'll be analyzing. So that is the first part which you have to understand before we jump into any type of indices. So the first question which we have to ask is, what is the raw data which we use in ecological analysis? What is the raw data which we use in ecological analysis? Sorry. So we could say that we could divide this raw data or the data which we are going to collect by sampling techniques into various different, levels. The most simple one is, where did it go? Oh, I guess I can't, draw on this and don't understand what's happening here. Anyway, so the most simple one is that is presence or absence. So if you have got, say, let us take a scenario where you are looking at two snakes. Say, let us say Tias mucosis. You all know what Tias mucosis is. It is the rat snake, Jera. And Naja Naja, that is our Murkha, that is king cobra, sorry, cobra. So you got just two snakes and you want to understand whether there is some relationship between their presence or absence or whatever you're going to study. So here the raw data can be at various levels. What are the various levels? The first level is whether it is present or absent, only that much. So you draw two columns and on one column you can write, rat snake, that is the heading. The second one you can write cobra, and then after that, present, absent, present, absent. So you may be looking at different times or you are looking at different spaces. Wherever it is, the only data you're entering is present, absent, present, absent. So you can convert this presence or absence data into numericals also. That is, if it is present, you can give a score of 1. If it is absent, you can give a score of 0. So this is the basic type of data which you will need in ecological analysis. I'm unable to find the, here it is. Thank God. Yeah. So all we need to do is you draw a column like this. This is species 1. This is species 2. And you may be looking at different times, maybe morning, evening, or whatever you are. You may be looking at it at different aspects, from different aspects. All you have to do is, okay, it is present in the morning. This is absent here. But in the evening, both of them are present. So this type of a data is called presence or absence. And here I just talked about 2 species. We may be talking about so many different species. Okay. And it need not be about species. It need not be about species. See, all the good ecologists need not be good taxonomists. Many a time, you may be only able to identify it to a higher level, that is maybe genus or family. So it may be like, you may be writing Colubridae, Lacertidae, something like that, because you don't know which species it belongs to or which genus it belongs to. You may be only knowing about you may be only saying that, okay, this belongs to this family or this class. But that is okay, but you'll have to write like presence or absence. The same in the case of, abiotic factors also. So whether there is sunlight is present, absent. Whether there is rain, present, absent. So this type of raw data can form the basis of various ecological analysis. So here we are only talking about presence or absence. And this can be indicated in numbers as 1010, where 1 is presence and 0 is absence. The second type of thing, or second level of raw data, which you look into, could be taxon or species count. So let us take another example. Say we are going to do a study at 3 sites. Okay. So one site is Viyur. I'm just matching V. The other site is Shaktan. And the third site is Puttur. So Viyur, there is a jail there. There may be some gardens or some sort of a place there. So that is Viyur. Then Shaktan Gardens. And then Puttur Zoo. And then here we are just looking at the taxon count or the species count. So here we are not talking about how many numbers there are, how many numbers per species there are, but here we are looking at how many species there are. So it could be like, say 7 species in Viyur, 2 species in Shaktan, 3 species in Puttur on day 1. On day 2, it is 3 species in Viyur, 2 species in Shaktan, 1 species in Puttur, something like that. This could be one way of looking at this type of data. Another way of looking at it could be at another level, where you are looking at families or higher taxa. So family 1, family 2, family 3. So we've got, different families, so many families are there, and from each family you have got, say, this many number of species or this many number of genus. So two species are there, one species are there, zero species, again three species, one species, one species, zero species, one species. It is present. Okay, only that much. So this sort of a thing is, it takes it to another level where you are actually counting the number of species or the number of taxon. So why I said taxon is because it is not necessary that you may be able to identify each and every animal to the species level. It is literally impossible for all of us to be such good taxonomists. So many a time you will need to get the help of another expert who is an expert in that particular group, genus or family, etc. At other times, you may be just able to say something like Rhacophoridae 1, Rhacophoridae 2, Rhacophoridae 3, which indicates that based on the morphological characters, you have divided them into three species and they all belong to the taxon Rhacophoridae. Okay, so that could be the way you're classifying this raw data. So this becomes very important. Why I'm trying to tell you this is because when you go out into the field, don't think you know everything and you're supposed to know everything, and you have to identify these are the mistakes which we people made 10-20 years back. Okay, so that is not the way things work. We need help, we need collaboration from so many people. So it's only when we get help from others, we will be able to get our data right. So that is quite, this part is very, very important. Okay, so if you don't know the correct identity, so it is better to give a species 1 or species 2, or say Colubridae or Lacertidae 1 or Colubridae 2, something like that, instead of giving a wrong identification. So this is another thing which is known as the count data, wherein you're counting the number of taxa. It could be genus, it could be family, or it could be even species. Okay. Or morphs. Another term which we can use here instead of species is morph. So sometimes when you go and look at different organisms, they may be looking like different organisms, but you are not sure whether it is a distinct species. But physically they are looking different. So then we are calling them as morph, M-O-R-P-H, morph. Okay. So this is the second level of raw data which you could use for ecological analysis. Then another thing which we can use about is the abundance. So here, again, it could be taxon level or it could be species level. So we could just recreate this same table here. So here, Viyur, Shaktan and Puttur. And instead of day 1, day 2, what I have put here is species 1, species 2, species 3, species 4, species 5. So after doing my sampling in these three sites, I was able to, identify 5 different species or 5 different morphs, and I have just named them like this. And then I'm counting how many individuals per species. So if you're using it for plants, we say how many stems per species. How many stems? Stems means, okay, individual. You don't use the term individual for plants. So you use stem, S-T-E-M. But for animals, we can always use this, that is species 1, that is 1, 2, how many individuals per species. So in Viyur, I saw 2 species 1. In Shaktan, I did not see any of the species 1. Then in Puttur, I saw 3. Then for species 2, in Viyur, I saw 1. In Shaktan, I saw 10. In Puttur, I saw 4. For species 3, 2, 2, 2. Then in species 4, 3, 7, 6. Then species 5, 0, 1, 0. Okay. So just looking at this data itself gives you a lot of information. So if you're looking at species 3, you understand that even though they are less in numbers, they are distributed almost evenly across all the 3 sites. When you're looking at species 4, you understand that they are also quite common in all these 3 sites. And for added advantage, they are in much higher number. So why these things are very important is when you start thinking about conservation strategies, etc., even these small things will be very important. So when you look at a species like species 5, you see that it is absent all over the different sites, Viyur, Shaktan, etc. But it is present in Puttur, etc. But it is present only in Shaktan. So what is the speciality of Shaktan, which is making species 5 present here? Is species 5 an endemic species? Is it an exotic species? There are so many things which we have to start thinking about species 5. When you look at species 1, here again the numbers are less and it is, more concentrated in Viyur and Puttur. It is absent in Shaktan. So there would be something special in Shaktan which is causing this species 1 to be absent there, or is it just coincidence? This has to be something which you will have to examine. When you look at species 2, you find that it is also present all over the 3 sites, but it is, concentrating in Shaktan, 10. So what is species 1 and species 2 antagonistic to each other? Because see, in Shaktan, it is very high and here it is 0. But that cannot be, because here it is present. It is present in both the other 2 sites. So both these species are present in both the other 2 sites, albeit in a lesser number. So is it something about the abiotic characters there? So many questions can arise just by looking at this type of data. So ecological data, whether it is presence or absence, whether it is count or whether it is abundance, will become very, very important when you start analyzing not only the community structures, the population structures, but also the interactions between the abiotic and the biotic environments. Then along with this, we may also have to measure various abiotic measure factors like sunlight, canopy cover, different types of temperatures like relative temperature, relative humidity, air temperature, soil temperature, water temperature, etc. All of these things, depending on what type of species or what type of ecosystem you are looking at, this becomes very, very important. Then with this type of raw data, you will be able to calculate a lot of things. You may be able to calculate relative measures, which is nothing but the percentage. So if you want to look at the relative percentage here, it is 2 plus 1